Right. Hello, everyone. Thanks for joining our FLOJO webinar on phenotyping. We will be beginning in about a minute or maybe two, so just sit tight. But as the webinar is going on, feel free to drop questions in the Q&A, which is in the center, and I'll try to answer them as we're going. We have a lot of people who registered for this webinar, so there might be a bit of questions, but I'll try and keep my eye on that. I might not answer questions if I'm going to answer them later in the webinar, but um, yeah, feel free to ask your questions in the Q&A. And yeah, we'll give it another minute and see if more folks show up, and then we'll get started. Okay, the time is 10:01, so I will go ahead and get started. Anybody else hopefully is on their way is officially late. But yeah, so let me go ahead and dive into it. My name is Dylan Hinson. I'm with the FLOJO Product Innovation Team. I'm a data scientist here at FLOJO. If you ever email flojo@bd.com with any question, it'll come to me or somebody else on my team. And so feel free to send us emails to flojo@bd.com if you have any questions after today's webinar or really about anything FLOJO related, or if you're stuck on something and want some help. We're always happy to answer questions if you email us, again, flojo@bd.com. And so today our webinar is going to be talking about phenotyping. And specifically, we're going to be focused on like trying to identify the identity or the phenotype of clusters that we receive from machine learning. So I have some quick slides. I'm going to try and go through fast before we jump into FLOJO proper. But here's some tools that we have in FLOJO that I find very helpful for phenotypes. We're going to focus on color maps, Euclid, Cluster Explorer, and marker enrichment modeling. And maybe we'll do some violin and box plots too. But all these tools are just ways we can kind of get into the nitty-gritty of figuring out what our clusters are and figuring out what their phenotypes are. Okay, so flow panels are getting big. In traditional analysis, in traditional high parameter analysis, we would see a bunch of gates is kind of how we define our phenotypes. And hopefully, most people on this call feel pretty comfortable drawing a bunch of gates. The challenge, of course, with drawing a bunch of gates is, how do we set those gates? How do we define them? It can be tricky, especially the more parameters we have, especially with some markers that might have dim expression. And especially with some markers that, a phenotype might be described by multiple markers. And so how do we account for the expression of multiple markers? So all these kind of questions lead us to, we still use, of course, traditional gating to verify phenotypes, but we also would consider using clustering and clustering tools. And so most of today's presentation is going to focus on, after we've done our clustering, how do we get the phenotypes? So clustering, is a useful strategy because it is an unbiased and objective way we can group ourselves together based on their expression. And a key benefit of clustering is that when the clustering algorithm looks at a cell to try and determine its phenotype, it's looking at all of the markers the cell is expressing. So instead of just looking at 2 dimensions and drawing a gate there, the machine learning algorithm is looking at every dimensions. That way, if maybe the cell has a little bit of an unusual expression for just one of its markers, it might still get included with its proper phenotype because it has the same expression for all the rest of its markers. So clustering is a very powerful tool that we have available in FLOJO, mostly through our plugins. I have a couple of clustering algorithms displayed here. FlowSum on the far right, I guess, XShift, and this might be Phenograph. But we have plenty of clustering tools available on our website. Again, those 3 I just listed, FlowSum, XShift, Phenograph, are probably the 3 most popular. If you're not familiar with using the clustering tools or using our plugins, I apologize that we're going to start with some already finished clusters for this webinar today, and I'm not really going to go into the specifics on how clustering works, but we do have a lot of other great webinars about clustering and about how to set up the clustering tools. So for today, I would just try and follow along and just know that, clustering, what that gives us is a bunch of populations as the result, and those populations represent phenotypes. So as long as you understand that clustering is returning populations, and the populations present phenotypes, you should be good to go as far as the presentation today, and you can always review our other webinars or our other videos to understand how to use our clustering tools and understand how to set them up in Flojo. Okay. So here's kind of what a high parameter analysis tends to look like. Step one is we cluster the data. We'll have to aggregate the data together potentially to cluster it properly, but we cluster the data. That's the first step. That will return populations for us, but those populations will be unlabeled if we're using unbiased clustering. And so we have to figure out what those populations are. The machine learning has grouped them together based on their similar expression. But it doesn't know what they are, like it doesn't name them. And so we might want to, visualize and call the phenotypes. That's step two here. And so we're calling those phenotypes based on, their names. And then, of course, once we have all of our phenotypes identified, we might want to compare different conditions, like a healthy and diseased condition, for instance, which I do have in this data set, but I'm not sure we'll have time to look at today. Okay, so if you imagine the whole high-dimensional workflow, there's like 9-ish steps to it. And those 9 steps might look like normalization, data QC, annotation, concatenation, dimensionality reduction, clustering, phenotyping, comparison, and isolation. So this webinar, we're basically just focusing on this step 7. And so you can imagine the first 4 steps are basically pre-processing the data to get it to a place where we can run dimensionality reduction and clustering on it. The next 2 steps are where we do our machine learning to kind of group the cells together and get their clusters. And then today we'll be starting on step 7, which is just trying to identify when I get my clusters represented here, what their phenotype actually is. And then steps 8 and 9, comparison, we're comparing different conditions, and then isolation. Maybe we're trying to bring that population that we've found that's unique back to the cytometer to then sort and get that unique population. Okay. So yeah, phenotyping is what we're doing today. So the first strategy I'm going to use for phenotyping is maybe the simplest, but also my favorite, to be honest. It's called multigraph overlays. This is a really cool feature we have in Flojo with any plot in the layout that doesn't have an overlay already on it. So any plot of just a single population in the layout, like this UMAP I have here, you can right click on it and you can choose make multigraph overlays, and you can choose make multigraph color mapping. And that will create all these plots, a plot for each parameter you choose. And so now these plots are basically heat maps of my UMAP here. So this is my UMAP results. Hopefully the UMAP has segregated my clusters based on their phenotype. And then we can see the marker expression in each of these clusters using the color map, the scales at the bottom here. The colors closer to the blue have little expression. The colors closer to the red have lots of expression. So green's kind of no expression, and yellow or red is usually expression or high expression. And so we can see just in a few of these markers here, like CD19 is clearly expressed up here mostly, CD8 down here, granzyme B mostly over here. So we can get an idea pretty quickly of what these cells' phenotypes are just using this multigraph overlay method. And I think the graphs are really pretty, and so I think it's a pretty nice first pass at how to identify our clusters, especially for the major phenotypes. We can pick those apart pretty easily using these multigraph color maps. I will pause right now just to remind people again, if you do have questions, feel free to drop them in the Q&A, but I'm moving through the slides kind of quick right now just so we can get to a live demonstration. Okay. So the next tool we're going to play with today is called Euclid, and specifically the clustr check function in Euclid. Euclid's a kind of neat tool in that it does two tests. The first one is cluster check, which is the one I have visualized here, and I think it's the one that's more useful for phenotyping. Cluster check checks the clusters to see if they have bimodality, to see if there's maybe multiple populations within the clusters, or if, in a parameter, if the cluster has different expression, like two separate expressions for the parameter, that might indicate that that cluster really should be two clusters instead of one cluster. So sometimes when we cluster the data, the machine learning algorithm will return clusters to us, but those clusters are actually still not, they're describing multiple subsets still. So within those clusters, there still might be multiple subsets. There's a few things we can do to try and get those multiple subsets out. We could always cluster the clusters again. We could always run clustering again with a different k nearest neighbor number, or if you're doing flow sum, just a different number of clusters you can return. So those are all options. And yeah, and so we can see here in this heat map here, there's a couple of places. In general, it's blue. That means that there's low modality, which means that the clustering is good. But in some places, there are these brighter colors, and that indicates that there's a split. For instance, in whatever cluster 1 is here, it has a split in its CD27 and its granzyme B and CD8 potentially expressions. So potentially, we could get further populations from this cluster. And also cluster 7 down here actually has a lot of just variance. It might just be because this cluster is full of outliers, but it might also be because it has a couple of different populations in it that need to get split apart. And so in general, I guess cluster 5, that's a very homogeneous cluster. That means that cluster doesn't need to be further separated. It's probably all the same thing that's in that cluster. Same with cluster 2. So this is just how you can read this, this cluster check results. Again, this is from the Euclid plugin we have available on our Flojo website. So it's half of what Euclid does, but I think it's the more important half for our phenotyping discussion today. Okay. Then the next tool we have built into Flojo now for for identifying clusters is Cluster Explorer. Cluster Explorer is probably the most straightforward way to identify a cluster, what it is, because Cluster Explorer gives you a few different visuals for looking at the cluster populations. You can see the clusters overlaid on the UMap. You can click on them, and it's very interactive. It also makes heat maps and line plots and bar charts. And the heat maps and line plots especially are useful for trying to understand, okay, this is maybe what our cluster's identity is. Okay. So again, there's just a few different visuals that Cluster Explorer makes. We can use it to filter for populations of interest. We can use it to backgate clusters. There's a lot of just different options built into Cluster Explorer that are really neat for visualizing clusters. Okay, then we have marker enrichment modeling. Marker enrichment modeling is also now built into Flojo. It's kind of somewhat of a new algorithm that we're using. But basically, it's just trying to identify the enrichment of marker expression within the clusters relative to the other clusters. So it looks at, the relative change in magnitude of the marker expression from our test population to some reference population. Oftentimes that reference population might be just the rest of the clusters. So we're comparing, the expression in one cluster to all the rest of the clusters to then see, okay, is it expressing significantly more of this marker? And if it is, we can then go ahead and say it has a high expression for that marker. It's going to have a high MEM score for that marker, and then we can kind of get an idea of what that cluster is. So MEM will return scores in a linear scale from -10 to +10. And so, if it has high expression, like down here, we can see some MEM results. This says HDLR is +10, HLA-DR is +10, so it has really high expression of that HLA-DR marker. But it has really low expression of the CD7 marker. Similarly, CD19 has a really high MEM value. So we can kind of get an idea of, okay, maybe this cluster is like some B cells just based on their HLA-DR, CD20, and CD19 expression. So we can kind of identify what this cluster is relative to the other clusters. Okay. There's some graphics here of comparing a test pop to reference pop. That's how MEM works. And so it's taking, there's two different ways you can you can do MEM. You can either reference a control population, so you can test against a control, or you can just reference against the whole rest of the cells. So you're taking cluster 1, referencing against cluster 2 and 3, and then you're taking cluster 2, referencing against 1 and 3, etc. I see a few questions. Yeah, we'll get into some of that. Um, so, so is there, so the first question going back to cluster check was, is there a certain number of cluster check value that's considered good or bad? And I don't have a value off the top of my head. I actually usually just look at the colors to try and identify um, where the splits might be. You can always look at the clusters like in your FlowJo plots as well to see if you think that there's actually a split there, or if that's maybe just noise, that like some variance that's coming from the noise. And so again, we can look at that more when we are doing our live demo. And yeah, you're right that if so it's, the cluster check has the heat map is kind of relative. So yeah, if there's one high score, it might make the rest of the clusters seem not as significantly different. Yeah, and then I guess another point that's being made in the chat is MEM does consider it's a relative, right, magnitude that's measuring. So it depends on what other markers you're including when you're running MEM. So if you're running MEM with a lot of markers, whatever marker is going to have the biggest magnitude of change, that's going to be scaled to the 10. So 10 is set at whatever the biggest magnitude of changes between two markers. And then for the rest of the markers, it's going to be relative to that. So if you have one marker that's really has a significant difference in its expression between its activated and inactive population, then that might wash out the MEM scores for the other markers, because they're going to seem like if they have less of a change in their expression between population 1 and population 2, then they're going to have a lower MEM score. But if you don't include the marker that has a really significant change in its expression, then its MEM score, and then the MEM score for the others will be higher, because again, it's a relative score. So we're weighting it based on the biggest change we see within the markers that were given to the MEM modeling. Right. So I might move on. I'm going to keep moving on just to try and get through, because we have a lot to cover today. Another thing that MEM can do, though, is it can rename your clusters with the expression of the markers that are within those clusters based on the MEM scores, so we can get an idea, maybe kind of a messy idea of what the cluster identities are. And then we can always go in and rename them based on what we think their phenotype is. Okay. And then the last thing about MEM is sometimes you want to adjust the threshold. So if I go back a slide, here you can see these names are really long because I'm including all the things that have +1 or -1 as their MEM scores, which means they only have a little change between the expression in that cluster and the other ones. And so a MEM score of +1 typically tends to not be a dramatic change. It might be within the noise of what we'd expect. And so instead of including the stuff with +1 for its MEM expression, we can filter that. We can adjust the threshold value. And so we can only show things with a MEM score of 2 or higher. That's going to get rid of a lot of the markers, and we can just focus on the ones that are really significantly different for these clusters. And that should help us identify what these clusters are and what's different about them than the rest of the cells. Okay, and then the last trick you can do in MEM is you can merge clusters together based on their MEM scores. And so if markers have if clusters have the same MEM score, it might indicate that it's the same phenotype or very similar phenotypes potentially. And so if we want to kind of group them together into their like major phenotypes again, we can always do that by merging based on MEM scores. Right. And so that's it for my slides in the slide deck, but I want to go ahead and jump into the live demo. Feel free to keep dropping questions in the chat or Q&A, and I'll try to get to them. So I did notice there's one other question in the Q&A that I see, and it's kind of questioning, if it's possible to automatically annotate the clusters. MEM is the closest thing we have where it automatically annotates the expression. But just because there's so much variance in what like a different cytometers, what the expression from different cytometers looks like, with the magnitude or the range of intensity values is for the cytometers. Depends on stains too. It's really hard to like use a machine learning algorithm, for instance, to automatically annotate clusters. It also depends on how people are naming their stains and what markers they're using. So at the moment, we haven't found like a great tool to automatically identify the phenotypes of flow clusters, though we've definitely looked at quite a few tools that get close. But for the process today, it's going to be up to us to name the clusters based on our knowledge of like the biological background. But we can get a quick idea of what the expression is, and that's going to help us a lot for identifying what these clusters are. Okay, so I think I answered the slide. Okay, so I'm going to go ahead and dive into FlowJo. In this workspace I have open here, we can see that I already have a concatenated file. It's a group of combined live cells from a few different samples that I want to run clustering on. I've actually already run clustering on it. So I ran some clustering on it. I ran UMAP on it already. So I'm already at the stage where I'm going to want to try to identify the phenotypes of the clusters that I got from my clustering. Again, if you're kind of new to clustering and haven't done it before, or you haven't used UMAP before, that's fine. For today's demo, we're not going to go into the background on how to do that, but we have lots of other videos on our website on how to do that, so I'd go refer to those. And for today, it's fine if you haven't done clustering before. Just know that the populations I have here are the results of clustering. The clustering has grouped my cells together based on their similarity in expression. And so what I'm trying to do today is identify why the machine learning algorithm grouped these cells together in the way that it did and what these phenotypes are, and if I needed to cluster them further to separate out further phenotypes, or maybe even merge them together if it clusters too much. And then a UMAP is just a visualization of my data. It's hopefully groups them again based on the similarity of the cells. So okay, so I ran clustering on this data. This data is a mostly T cell panel of a PBMC. It has a bunch of activation markers in it. I got this from the Flow repository, and I think we're going to share this data after the webinar. So if you want to try it yourself, you can watch the recording and hopefully follow along. But for now, we'll look at this together. So when I ran the FlowSum clustering, there's we have a few different clustering algorithms available as plugins in FlowJo. If you're looking for the plugins, you can find them under the Workspace tab, plugins, and then they're on our website on the FlowJo Exchange. So if you click on that, it'll take you to our website, and you can always download the plugins from there. FlowSum comes in the plug and play bundle, which is the easiest way to set up your plugins. So I would encourage people to do that. And what's unique about FlowSum is you can set the number of clusters versus X-shifted Phenograph will just return a number of clusters. They're kind of unbiased or unsupervised, so they'll return a number of clusters that they determine, versus for FlowSum, we can tell it a number of clusters, and that's how many clusters it will return. So I told it to return 7 clusters for this data set. That's a pretty small number for PBMC. It's going to get me all my major phenotypes, but it's not going to get me all my different activation states, for instance. And I kind of knew that. I'm doing that because I'm doing a clustering, or I'm doing a phenotyping webinar. It might be a good strategy if you want to get all your major phenotypes first and then look at all your activation states second. You could start with a lower number of clusters like I did just to get the major phenotypes. If you do this strategy, though, you'll probably want to cluster again. So for instance, I got my major phenotypes here, but my T cells are probably just split into CD4 and CD8, and they're not probably split to naive versus memory or other activation states. And so I'm going to have to cluster those data sets further, those populations further, to get the further subsets. the alternative would be we could run something like XShift or Phenograph. It would probably return 30 clusters to us. And I think that's fine. It's just we then have to go through and identify each of the clusters. It's going to get all the different subsets probably. And so we'll just have to go one by one through each cluster to identify what those subsets are. The first thing I would do to try to understand what these clusters are, because obviously right now they're just labeled 1 through 7, and I don't have a lot of information about them, is I would open up the layout. And in the layout here, I've already done two things. I've added my UMAP plot, and I've done a overlay. So I've dragged and dropped these clusters onto the UMAP, and you can see the colors now indicate, the different populations. So let me go ahead. I'm just gonna remove the sample name from this plot since they're all from the same sample. Who cares? Okay, so now we have the different clusters here. We can see 1 through 7, and you can see them on the UMAP plot. And I realize this might be kind of small, so let me go ahead and make it bigger for you all. Okay, so pretty quickly we can see the clustering in the UMAP kind of match fairly closely. I would say it's a pretty decent job. There's clearly some populations that have separated and aggregated together. For this one, there's maybe a few clusters that were included here that have some differences from each other. But for most of the populations, they're pretty much one population that matches between the UMAP and the FOSSUM. We also see some pretty big clusters down here that might have multiple subpopulations within them. And down here, there's kind of a curious thing because it's included, this is a separate cluster, even though on the UMAP, they're very much kind of together on this island. And there's a couple maybe outliers or different phenotypes here that it maybe was kind of struggling to place or put close to this island as well. So I guess, how do we identify what these are? Step one, and I think the easiest step, is we just go back to the original UMAP that I put in the layout here, right click and say make multigraph overlays and make multigraph color mapping. That will open up this parameter selector. And in the parameter selector, I can go ahead and choose my stains of interest for identifying the phenotypes. This panel is pretty big. It has maybe like 30 or 20 something stains. And so I'm not going to want to use all of those right now because a lot of these markers aren't going to be useful for identifying the major phenotypes. So I'm just going to stick on markers that describe the major phenotypes right now, like Granzyme B, CD4, of course. Let me see, do I have my CD8, CD3, CD19 is a good one. And I might be missing some, and maybe like TCR gamma delta, because those might separate really cleanly. HLA-DR could be useful. I hit the wrong button on my keyboard, so unselected everything, which is unfortunate. I'm selecting these by holding down the Control or Command key. I'm on a Mac, so it's actually the Command key on the keyboard and just clicking, and that will allow us to select multiple markers. So Granzyme B, CD4, do I have everyone? So these should describe my major phenotypes, CD3. I could maybe, I could have included some of the naive, like CD45, RA, or other markers as well. But for now, I think they're mostly going to separate based on the major phenotypes. So you'll notice the panel didn't have things like CD56 or CD14. And so, it's very much a T cell panel. It's mostly focused on the T cell expression. And so we're not going to be able to really get into all the subsets for like the monocytes or, the NK cells. So for that, or even for the B cells really. So for that, we're maybe just going to try to group them initially, but then ignore them for the rest of the analysis. But we can see here in this heat map that is returned, the different markers that I selected, and we can get an idea of what my clusters are. So I'm going to move these closer to the clusters so we can kind of see them together. I'm going to make my layout really big so we can fit everything on here, and hopefully it's not too small for everyone. And so we can see here, just going through CD19 expression. I'm colorblind, so you might have to bear with me on some of the heat maps here, but I think it's pretty clear that the CD19 expression is mostly concentrated up at the top here, where it's the orange or reddish color. And then there's maybe some CD19 expression down here as well. And it looks like maybe a tiny bit over here, but mostly up here at the top. And so I'd feel pretty comfortable saying this top cluster, those are probably our B cells just based on CD19 expression alone. And again, I don't really have much of a focus on B cells in this panel, so I'd feel comfortable just going in, renaming this top cluster our B cell cluster, and then ignoring them for the rest of the analysis. I think that top cluster is cluster 5, but I'm colorblind, so I always double check by changing its color. Okay, yeah, and then changing it back. And so that is definitely cluster 5. And so I can right click on this, rename it, call it maybe B cells or just CD19, maybe positive. And then leave it at that. Those are our CD19 positive cells. So with the TCR gamma delta, we can see pretty quickly, and maybe let me zoom in a little bit just because I feel like this is going to be too small on your screens to really see in a meaningful way. The TCR gamma delta population is mostly, the expression is concentrated very deeply right here. You can see there's very high expression here, and then kind of medium expression here, and then everything else has low expression. So this is definitely our TCR, our gamma delta T cells. And it was included though with whatever the rest of the E's are, which are hopefully another type of T cells. So we'll have to split them apart later. We'll have to do more clustering. So let's keep on looking at these other markers. Granzyme B is located here and here, and on our TC and on our TC the gamma delta T cells. So this is indicates maybe NK cells or maybe some CD8 cells. And so one of these is probably CD8, probably this one on the right, and the other is probably our NK cells. And again, since I don't really have other NK markers in this panel, we're probably just gonna call this population the NK cells and leave it at that. But just to make sure these are the NK cells, let me look at the expression of some other stuff. CD3 and CD8 are negative for that population, which indicates to me that this is probably the NK cells. You can see CD3 and CD8 are also super negative for that top population, which really confirms that they're probably my B cells or CD19 cells. CD4 is also super negative. HLA-DR is super negative. So since I have no other NK markers, I'm guessing these are mostly my NK cells. And again, if I was doing a study where I actually was interested in my NK population, I might have more markers to really confirm that. But in this case, I'm just going to go ahead and call these my NK cells. So this population we have to find on our map again. It's this orangish color, which I think it's maybe this one. I'm going to change the color though, just to double check. It sure is. I might make it a different color. I'm going to make it black. And that was population 2, so I can rename it NK cells. And I should note, as we're going along with this phenotyping webinar, we have a lot of immunologists who work here at FlowJo, but I'm not really one of them. I come from a bioinformatics background more so. I'm a data scientist. So some of my phenotypes I'm going to call very generally. And I know that we have very specific definitions that are often debated about some of the phenotypes. So bear with me if I call something an activated population and it maybe there's more nuance than what I'm really describing, or I called that an NK population, but I just use granzyme B. Obviously, we can maybe use better definitions to define those cells or more markers. But for this webinar, I think we're just trying to get the broad idea of what these populations are, and we can always verify it further with looking deeper at the marker expression. Okay, I see some questions popping up. Let me just check. Somebody asked if it's possible to include different like categorical factors when doing clustering. So for instance, like a patient ID or a treatment or a condition, I wouldn't give them to the clustering algorithm. Like I don't want it to separate my clusters based on the patient ID, for instance. I want it to have the same clusters for all the patients. But you can then, after doing the clustering, you can split the concatenated sample apart based on its patient ID, which I have included some of those types of annotation in it, which we could then use to split apart the concatenated file and apply the same clusters to the multiple different patients to see the same clusters changing between patients. And again, that's kind of in our clustering webinar, which you can go check out. But we're not going to look at too much detail today. Okay. And then can we make the cluster name appear on the UMAP was a question. Not by default, I guess this is the short answer, because it would get really crowded, especially if we had like 30 clusters. But my suggestion, my trick is always I just make a text box, and you can always call it like, we just looked at the NK cells, for instance. Sorry, my computer is being slow. NK cells. And then I always like to just make the text box transparent. So you can go over to here to fill, make it transparent, go over here to the line, make it no line. And then, whoops, I had the plot selected instead of the text box, which I didn't want. I want the text box. No line, no fill. So now I have this NK cell label. Oh, my NK cells are black, so I should have thought about that. But you put labels like this, and so then I could go up here and do that same thing with the B cells. It's like a manual method, but it tends to work the best for labeling our clusters if you want to label them. So I would just do that. Okay, continuing on, I want to get some of these different clusters identified. Again, I'm just going to be using broad strokes with these heat maps, but I think it makes it pretty, I think it's pretty obvious with these heat maps what the major phenotypes are. CD3 is super expressed here on the right side of the graph. It's basically what the difference between the right and left side is the CD3 expression. So these are our T cells on the right. We've already seen the gamma delta T cells are there. Based on CD8 expression, we can see our CD8 is at the bottom. Our CD4, therefore, is probably at the top, which we can see over here. So, broad strokes, we can tell these are my CD8 T cells. These are my CD4 T cells. And we can go ahead and rename that over here. Now, this is a T cell panel, so obviously within those T cell populations, I'm going to have a lot of different subsets. And so we can then cluster these further to get all the different maybe activation states. So let me just call this CD4, and let me call the bottom one CD8, which is, sorry, I'm going to have to change the color again so I can try and understand what this is. I'm going to go to like a blue. Okay, that's it. And then I'm going to go back to red. So, oh, I changed the NK cells, my bad. Change this to red, and I'm going to change this back to black. Okay, so those are my, so cluster 1 is my CD8. Okay, I'm already kind of regretting one thing I did. So as I'm renaming these, I'm just doing it so I can get an idea of, what these are, and then I can cluster them further. But I actually maybe want to keep their cluster numbers just because some of the tools we have in Flojo are going to use the cluster number rather than use the names I gave it, or sometimes they use a combination of the cluster number and the name. So it might just be useful to know what the cluster numbers for these populations were originally. I can tell what the cluster number is because of the order that it is in the, I went from the lowest number to highest number in the heat map here. So for instance, this is CD4's cluster 3. So I'm just going to make sure I put the cluster number out front, just so when we look at some of the visuals later, we understand what we're looking at. This is cluster 5. Oh, I hit delete. You can undo that. Okay, so I am 3 populations away from naming all my major phenotypes. Which ones are we missing? Okay, let's look at this big guy on the left. So I don't have any monocyte markers. While I don't have any of the classical monocyte markers. This isn't a panel that's targeting monocytes at all. But we can see over here on the left, this has a lot of HLA-DR expression. So I'm betting just because my monocytes are going to be missing here and they're going to be kind of negative for all the other markers, that they're big cells, so they have like this dim expression for all the other markers. But I'm betting that on the left here, these are my monocytes. They might also so I might have a few cell phenotypes that I didn't really label with this panel that might be kind of getting grouped in that big population over here. And in fact, I think that's what's happening with these other two islands that are being included with this one. So really, this cluster 4 here is like my unstained, or it's like the things I didn't target within the panel. And so I'm just going to call it my unknown or my everything else. It's kind of the cells I decided I didn't care about when I was designing the panel. I actually didn't design this panel, but when the person who's designing this panel chose the panel, they weren't targeting these cell types. And so we really can't say a lot of, or shouldn't really investigate the cells that are in here. Just know we've separated them out, and so they're not going to be mixed in with our T cells, for instance. Okay, so I'm just gonna call that unknown, and let's just leave it at that. They're probably the monocytes, as well as maybe a few other cell subsets that are mixed in that weren't really stained or targeted in this panel. And then, okay, we have these blue cells and this guy, which I am gonna change the color of to kind of get an idea of where it is. It looks like this is mostly, it might be an interesting population. It might just be outliers. I'm going to rename this outliers for now. You can always look at the marker expression to see if it's some interesting subset that, has very few cells. Maybe it's a rare subset. But it might also just be a group of the outliers that had irregular expression, and the machine learning algorithm just grouped everything with irregular expression together because it didn't know what to do with them. And then last but not least, we have this kind of purplish population. It's clearly CD8 positive. It is CD3 positive as well. So it's some type of CD8 T cell. It might have some enzyme expression. I'm not really sure why my machine learning algorithm grouped this so separate from the others. It has some HLA-ADR expression. It's just a smaller CD8 population. If I looked at the activation markers, I'm sure there was some significant difference about its activation state than my other CD8 cells. But for now, I'm not looking at the activation markers. So let's just go ahead and rename that. I'm going to call it small CD8. And I already forgot that I wanted to label these based on their cluster numbers. So that's 7. And this one's 8. And so my small CD8 population, I might want to merge back with my big CD8 population just for the sake of getting my major phenotypes together. And then that way I can run machine learning on that small subset, or the CD8 subset again, to further subset it. So let's go ahead and do that. Let's merge my small CD8 population with my big CD8 population to just get all my CD8s together. I can do that by going to Tools, Make OR Gate. Okay. And so now we have this combined gate here. Again, if you're not familiar with the Boolean tools, they can just combine populations together or separate them using the Boolean mathematic logic. So I combine my CD8 cells and my CD8 or my small CD8 together to make this combined population. You can see it's just the two of these populations added together. And I'm going to rename that and just call it total CD8. Okay. So I've got my major phenotypes right now. I'm going to take a look at the chat for some questions. We have, okay, cool. I think I'm done with all the questions. So the next step I might do is a few things. The first thing let's check out is I mentioned that within these clusters, there's probably going to be a lot of modality because we haven't looked at any of the activation markers yet. But the way we can actually check that is with Euclid, which I kind of mentioned in my initial slides. So Euclid is a plugin available on the Flojo Exchange. Again, we have lots of videos on our websites about how to set up plugins. And if you download a plugin, it comes with a how-to PDF that explains how to set up that specific plugin a little bit. But I'm going to go ahead and run Euclid. For Euclid, we want to consider what markers we're giving, all of the machine learning, everything we're doing, of course. The only thing I'm going to remove for the Euclid run is the stains I've already used, and the only stain I've already used to get to my population here is the live dead stain. So everything is alive in here, so I'm going to remove live dead. And then for the categorical parameter, we'll just want to choose our clustering parameter, the flow sum, and we can go ahead and run Euclid. Euclid does two tests. It does the cluster check and the Taylor index. We're mostly going to be interested in the cluster check today. The Taylor index, just to let you know what it is a measure of the kind of like how much the clustering matches the distance between the populations in multidimensional space. So it kind of is evaluating how good the clustering is a little bit. So you could always, if you're curious about the quality of your clustering, you can run Euclid, you can get the Taylor index, and the higher the Taylor index is, the better the clustering is. Might double check that in the documentation. The cluster check, though, which is what we're curious about today, and it just got returned to us, it should show us the bimodality within the clusters. So here we go. So it returns this graphic here in our layout. And you can see, let me make it bigger, I guess. You can see each of the clusters here on the left. Again, it's just using the cluster numbers. That's why I went back and I relabeled these with the cluster number, although I definitely labeled it incorrectly. I skipped 6. So it should be 6, I think, and this should be 7. Okay. 1, 2, 3. Yeah. Let me just double check I got the numbers right. So 1, 2, 3. Nope. Unknown is 4. It should be in the order 1, 2, 3, 4, 5, 6. That should be 7. Okay. Okay. So going to Euclid now, these numbers should match. So we can see pretty quickly, yeah, we expect that some of these clusters have a lot of modality in them. Cluster 1 does, that's our CD8s. We saw that with the CD8s. Cluster 2 does not, and that's our NK cells. So that makes sense because, again, there's not many NK cell markers in this panel. There's basically none. So they're not going to have a lot of modality because we didn't stain for any of the NK subsets. CD4 has some split, but CD45RA, so there's a strong split within the naive in memory there. The unknown, yeah, there's some split there. 5 is CD19. Again, we didn't stain for its subsets, so it's basically homogeneous as far as this panel is concerned. We're not going to look at any subsets for it. We're going to do no further clustering there. And then so number 6 is just CD8 again, so that has some modality in it. And then number 7 are the outliers, and of course that's going to have super high modality. So that makes sense because, again, the outliers were kind of all over the place. So okay, so what can we do with that? It can inform us what we should do more clustering on. And let's do more clustering on our CD8s. I have this total CD8 population here we can do more clustering on. Because I want to get all the different subsets for my CD8s aggregated together. I even have my gamma delta T cells mixed into this CD8 population, and so I want to split those out for sure. So let's run some clustering on our CD8s here. This time for clustering, I'm going to run XShift just to do some unsupervised clustering. So I'll let the machine learning algorithm decide how to split these. We know a little bit about this population, so I don't really want to include markers that aren't going to be useful for the clustering. And so some markers that for sure aren't going to be useful is CD4, since these are CD8s, they should, for the most part, be CD4 negative. CD8, obviously, we've already used that. They're all CD8 positive, more or less. We've already used live dead. They're all CD3 positive. So we really only want to include things that might be changing between the different subsets that might be useful here. So the rest of these seem, yeah, they seem okay. So I'll go to include the rest of these and run the clustering. It is 60,000 cells, so it's going to take a moment. So now is a good time to maybe answer some questions. Yeah, so one question was, how can we phenotype the outliers further to make sure they are indeed outliers? Again, you can use all the tools we're going to be looking at today to do that. So we're going to be making some heat maps later, as well as we're going to be using Cluster Explorer. And all those things can tell us the marker expression. We can also always just open up the subset in a 2D plot and just look at like some of its expression here. For instance, maybe just HLA-DR. And we can see its expressions kind of all over the place because I think they're outliers. And then there's another question in the chat. How do you separate your gamma delta T cells? And we're doing that right now, I guess is the answer. So I'm going to separate the gamma delta T cells and all the major subsets by doing the X-shift clustering, which just finished over here. So it returned 19 clusters to us. So it has separated all of our subsets probably. And we could go back. I'm going to make a new layout. I'm going to call this CD8 and just focus on CD8 here. We could go back to our CD8 population and use that same UMAP that we had before to visualize the data. This UMAP, of course, just includes the things that were included in my CD8 cluster, which isn't much. And it's going to be really tight in the bottom right-hand corner. And so we could overlay them just to get an idea of what it looked like compared to that past visual that we had. And we can see it's split, this into many, many different populations, and it's done maybe some decent work compared to our previous UMAP. But this is kind of a hard visual. I mean, this isn't well spaced out. We can't really see what's going on here. So I might run a second UMAP here just to kind of separate my populations a little bit better. When I run this second UMAP again, I'll just want to exclude things that aren't going to be useful to seeing differences within those populations. So probably those, that seems good. I'm going to run the UMAP, should finish somewhat fast. Yeah, while that UMAP is running, more questions. Now's a good time for more questions. We have 8 minutes left, and I have a lot more I want to show. So hopefully it finishes quickly. Unfortunately, all the machine learning algorithms do take a moment to run. And while they're running, it's usually, you can do some minor analysis. So for instance, we could make some really bad UMAPs over here with using this little one. And I could try to, look at the expression again. Okay, here we go. Here's a much bigger UMAP of our CD8 cells. So this will be much more useful. It actually almost kind of has the same shape as this little island here, which kind of makes sense because it's the same populations. I bet without looking at anything that this thing on the right is our gamma delta T cells. And then somewhere in here we have the granules, the one with the granzyme B and the population without. So let's take a look. I'm going to duplicate this really fast, holding down the Option key on my keyboard, clicking and dragging will duplicate. And so the reason I duplicated it is because this bottom one, I'll go ahead and put the clusters on top of. And so we can see that it's split apart into many, many, many clusters. How good those clusters are, we'll maybe figure out in a second here once we start identifying what these clusters are. Let me go ahead and, oh, I made my layout really big. That's why it's not letting me scroll. Okay, there we go. So I'm going to right click on this, do the make multi-graph overlays, multi-graph color mapping. I like to start with this just because I think it's the easiest way to visualize some of this stuff, and it should be pretty obvious what our gamma delta T cells are. For this multi-graph color map, we can include many more of the markers because we actually care about the activation markers this time. I'm not just trying to get my major phenotypes. I'm really trying to describe everything or get some idea of what this populations are. And so if we include everything this time, it's going to make a lot more plots, and it will take a moment to make them, but here it is. And so just to kind of hone in, okay, CCR7, CCR7, obviously it's super expressed on the left side here. So we can already get a pretty fast idea of this tetramer that the panel includes. obviously this population is that tetramer. And so we can get a pretty quick idea of what some of these things are. And so let's go ahead and just rename this MR1 tet is apparently this top population here, which actually has two things in it. And it is a population I'm going to struggle with a lot as a colorblind person, so I might just skip it for now because it's too similar to the other red colors. I don't really understand. The TCR gamma delta is the thing I'm most interested in, and it has been it's in that right-hand corner. It's that one I kind of pointed out before, my gamma delta T cells. And so it made a bunch of different clusters that are gamma delta T cells. I could try to go through and visually identify which clusters are which and group them together. I could also, though, use another strategy. And so I only have 5 minutes left, so I'm going to try and use some of those other strategies. The first thing I'm going to do is do Cluster Explorer. Cluster Explorer is built into Flojo now. It's this button here under the Workspace tab. In the newest version of Flojo, you'll have to use the newest version if you want to see this. For Cluster Explorer, again, I just want to include all those markers I was using before, which are the ones that are changing between these different CD8 subset populations. That should be pretty decent. And then I want to include just the UMAP for my CD8s. So we should pop up with Cluster Explorer here in a second. Cluster Explorer brings a lot of windows with it. It's taking a second. Here we go. So it has a lot of different visuals. One of them is actually hidden right now. It's behind this guy. Right. So it has these 5 different panels, 4 different visuals. This one on the right is just a bar chart. We can probably ignore that. This 2D plot is pretty neat. This is the UMAP that we were looking at before. We can click on populations to see what cluster they are. So this is cluster 7. You can see its expression. This is a line chart. It's showing the expression for these different markers. And so we can get a pretty fast idea of what this population's expression is of all these different markers. So I can see, for instance, it's CD27 positive, CD28 positive, CD127 positive, and gamma delta T cell positive. And so that tells us pretty quickly that, okay, maybe this is some sort of naive population. And then we can go, but it's definitely the gamma delta T cell population, which is why I was kind of interested in. And so we can click on these different ones and we can maybe see some changes. For instance, this one, which is a gamma delta T cell population, was CD127 positive, or it had highish expression of that. This one has much lower expression of the CD127 marker, and it maybe has some expression of granenzyme B. So we get an idea of why it's separating these different subsets into clusters. But we can see, the through line, all these ones definitely have that TCR gamma delta expression. And so if we want to group them together, there's a few ways we could do this. One is we can draw gates here and try and group them just based on the expression of some of the gates that we're drawing. And so now you can see we've just got all of our potentially gamma delta T cells grouped together. And so that's one tool we have. We can always merge the selected clusters together if we want to merge them together in the workspace. If I'm feeling brave, I could do that. Sure, let's go ahead and do that. So do I feel like I got it? Yeah, I feel like I got it. And so now it's merged them together back in my workspace. Whoops, that's not my workspace. This is my workspace. So it's merged those gamma delta T cell populations together. Oh, it won't maybe make it though until I close Cluster Explorer. Just to show one last thing in Cluster Explorer, we have our heat maps here. If I'm interested in a single population, I can click on it. It'll show it in the heat map. The heat map, again, you can see the color scale on the right. Red is high expression, blue is low expression. So the population that I selected here in the bottom right, I have so many parameters that the labels are a little bit cut off, but you can see it's CD45RA positive, and maybe granzyme B positive. These expressions are all relative to each other, so it's I mean, some sort of naive population probably. But you can get some idea of what the expression of these different populations are by clicking around in here. And you can always, somewhere under here, you can export the graphic, but you can also copy the content, which will copy the values, the median intensity values for each of these clusters, for each of these parameters, and you can put it in Excel. Okay, so I have a minute left, but I have at least one or two more things I wanted to show. So I'm gonna go ahead and just maybe take some extra time. But if you have to leave at the top of the hour, I definitely understand. Thank you for joining me today. This webinar will be on our website, so you can see the last couple of things I'm gonna do here if you watch the recording. I think they might be including the data as well, so hopefully you can get access to both of those things. All right, let's go ahead and close Cluster Explorer just so we can get that gamma delta population that I merged. Should have made it, but I don't. Oops. Don't see it. Maybe I didn't press the button, right? Okay, let's just carry on. I don't know where that population went. Did I not press the button? Anyways, I don't know where the population went. Let's just carry on. Okay, so another trick we can do is we can use Violin Box Plot. So under plugins, we have this tool called Violin Box. It makes some simple-ish heat maps and Violin Box plots in R. I like the Violin plots, so let's look at those. If we include too many markers, it's going to take a little bit of time to run. So I definitely want to remove the ones that are not as useful. There's some other markers, though, that I just remember for this panel weren't super helpful. Like the PD one didn't really work. I don't remember what else was not very useful in this panel. Okay, let's just run with these. And then we'll go ahead and choose my X shift and then open up the Violin Box Plot plugin. It might, here we go. It'll open it in a new browser window. It'll show us all the median expression values for all of our clusters. I'm going to bop over here, though. It can make some heat maps. It should make this pretty fast. So let's go ahead and see. I already want to change this. So the heat map by default has a dendrogram, and you can see it has each of the clusters here with their different expression of these different markers. It's not scale. It's based the heat the expression the heat map color is based on the expression of it's using the same range for all the markers, and so if one marker has high intensity values, which I guess CD95 or CD45 RA does, since CD45 RA has such high intensity, it's making everything else look like it has low expression. So I'm going to normalize everything by for each column. And so we can see within each of the columns here what has high expression for these markers, what has low, what has middle, and just get a better idea of what the differences are between each of these clusters. So now, for instance, we can tell, let's look at the, we still have our gamma delta T cells should separate pretty nicely. So the 16, 11, 10, those are definitely gamma delta T cell populations. And I think even maybe some of these other ones. This is a hard colors for colorblind people, but you can always change it. And there's a lot of different options in the Violin Box Plot plugin to make these visuals. So something like this can help me see a little bit better what the different populations are. Okay, and then CD45RA, we can see here's our CD45RA positive populations, and then these ones are all negative. So it gets us some idea of the identity of these different populations. We could always go back and rename some of these or group them maybe based on their expression of these markers. But this is one way to phenotype. Okay, another, we can make these Violin plots along with jitter and box and whisker plots, which I'll do. They take a second to make, and I'm including a lot of parameters. So this could take a minute. While this is taking a moment, let me check the chat. Yeah, somebody is asking about the median values if you copy and paste from Cluster Explorer. I think they're a bit off the newest version of Flojo, and we haven't released a new version yet that's going to fix that. Um, you can the same normalization by yeah, the Cluster Explorer heat maps. I think the scale of them, I'd have to double check, but I'm pretty sure they're using the same scale of the parameters that are in Flojo. So for a lot of these tools, whatever the transform is that you have set for the parameters in the data set in Flojo, we're going to use that transform for the color maps, for instance, that we're making in the layouts, and other tools as well. Like MEM will use that transform as well. And so you want to make sure you have a good transform set for your data before you use machine learning or any of these phenotyping tools. And when I say a good transform, I mean you want to try and separate your positive and negative population using the transform as much as possible. And you really don't want to leave white space on your scale. The violin plots returned, but before we look at that, let's just go ahead and look at transforms real fast. So like good, I've transformed all this data so you can see we have good separation between our positive and negative populations, more or less. What we don't want to see is something like this, where we have a lot of wasted white space that we're not using. So we definitely want to make sure we don't have that white space that we're not using, and we're separating these positive and negative populations as much as possible. Okay. These violin plots returned. I have really long names for all my clusters. Once we rename these, it's going to look better. But we can get some pretty fast understanding of the different clusters and their different expression for these different markers. For instance, CD45RA, we can see these ones have much higher CD45RA expression than these ones down here. So it gives us some idea. I guess there's a lot of background. For the CD45RA, these are still some middle expression, but it gives us an idea of what these different clusters are. We can see that gamma delta T cells, again, are really standing out. These 6 are definitely my gamma delta T cells. The rest are not. So we can get some understanding there. Whoops, I have 2 tabs. I don't really want 2 tabs. Okay, I'm gonna go ahead and close violin box plots. There's a lot of other things you can do to adjust the violin and box plots, but I'm gonna leave it as is for now. I actually tried to add the jitter in box, but I think I have to hit refresh in order for that to actually take effect, and it's gonna take a while to calculate. So let's close this for now. If I wanted to clean this up, I would rename these, which there's actually a button to rename the axis here, but it's going to be easier than renaming the axis in here to actually just go over to here and rename the axis in Flojo and then rerun violin and box plot with renamed clusters, because the new cluster names should go into violin box. Okay, the last thing that I wanted to show was MEM. And so let's go ahead and do that. I'm looking at my checklist, and I think we've covered most everything else. I haven't really renamed any of these populations yet. Maybe I should do that just to kind of demonstrate. The easiest ones to identify are, of course, these 2, which are both the MR1 tetramers. I kind of got scared because I'm a little confused about what colors these are. So let me just, nope, that's not it. Maybe it's this one. Make it blue. Nope, still not it. This is the colorblind person struggling with a limited color palette. I'm gonna make this black. That's it. All right, so it is CD4 or cluster 4. So I'm gonna rename this. I'm gonna keep the cluster 4 part. I'm gonna call this MR1 tetramer. So that's what that is. And then we have that second one here that is almost an even harder color for me to see. I'm gonna guess maybe it's this one though. I'm gonna make it like a gray color. Yeah, that's it. Okay, so it's cluster 1. So these 2 are my MR1 tetramer clusters. Whoops. And I actually am noticing, whoops, I must have pressed the right arrow key on accident. because I accidentally named it unknown. I want to name it MEM. Okay. So just to show what it looks like to have two of these maybe differently renamed, I would want to go through and do this with all my clusters using the expression here to get an idea. For instance, in my gamma delta T cells up in the right here, I can see they're split on their CD45RA expression. So we have naive memory, which I might want to mark. So I could go through and rename those individual populations. But since we're past time, I want to go ahead and do some MEM just to show how to use it. MEM is going to make some really long names, but marker enrichment modeling, it will create MEM scores for me based on the change in magnitude of the expression of whatever markers I give it. I'm just going to want to give it markers that will actually be changing between these different clusters. My PD1 stain didn't work. Let's see here. That's probably good. Let's go ahead and press OK. MEM is going to take a moment to calculate. I see one question in the chat. Yeah, there's some questions about the Cluster Explorer heat map. I'd have to look to see how we scale the heat map. I can't remember off the top of my head. It might say somewhere in Cluster Explorer. It definitely says in our documentation. Okay, so MEM returned results. It returned too much results though. The names of these populations, MEM changes the name of the population to have the MEM scores there. So we can see it ranked it from highest to lowest. So for instance, for this MR1 tetramer, I must maybe didn't include MR1 because it's not listed here, but oh no, it's down there. But it has CD45RA negative expression, so it's actually not CD45RA positive. But it does have expression of CD28, CD161. We can get an idea of what's being expressed in these different clusters, is really what we're showing here. For instance, this one has granzyme B expression, but it's CD45RA negative. Obviously, it's maybe too many parameters for us to really be able to read this full name and comprehend it. So to clean that up, we can change our MEM threshold. A MEM score of 1 maybe isn't so significant. I kind of want to look at things with a higher MEM score because that's what's really going to describe these clusters. So we can change the MEM threshold. If we go back to MEM, hit change MEM threshold. I can change it from 1 to 2. I can go back to 1 at any time. But now we've cleaned this up so we can see the different expressions here. So for instance, this is one of our gamma delta T cell ones. We can see it's gamma delta T cell positive, granzyme B positive, CD45RA negative. And so we can get an idea of, okay, I think all these first ones are CD45RA negative. So there may be memory populations versus the lower down ones. Like this one, for instance, is CD45RA positive, Ki67 positive, CD27 positive, CD28 positive. It has really high MEM scores for these. So it's definitely, some CD28, CD27, CD38, TIM3. So it's some naive population that's going into activation. And then down here, we have a gamma delta T cell population. So this is just how we can look at these MEM scores and try and identify, okay, gamma delta T cells, granzyme expression, Ki67 expression, CD45RA, super high expression. So we can get an idea of what this population is and maybe what's unique about it and how we would describe it. And again, once I've kind of figured out the identity of this population, I might not want to leave these MEM scores here because the names are really long. And so it's going to look bad. Like already it's kind of made my layouts look pretty bad because it made the names really long here. So as we figure out the identity of these, I would just go ahead and rename them. So for instance, let me find something with really clear. Okay, so this has, so this is some naive population. I'm just going to call it CD8 naive. We can maybe get more into the nuance of what this actually is. Oops. And then something like this is a gamma delta T cell population. It's also some sort of naive gamma delta T cell population. So I could rename it T gamma delta naive. Um, this is probably also a naive population. So we could kind of make calls like this. This is probably some sort of CD8 naive and try to identify them this way. Again, you can get more into the nuance of what markers are being expressed specifically in this population and what that means as far as the activation state of these cells. Oops. And again, I probably want to leave the cluster number just because otherwise it's going to get confusing later where these clusters came from. So let me do that for each of these. Okay. With that, though, I think we've gone 15 minutes over. We kind of went fast through a lot of stuff today. I'm going to hang around for a while. If you want to ask questions, feel free to drop them in the chat. Um, yeah, this is the end of the webinar though. So we covered everything I wanted to show today. Hopefully this gives you some idea about how you can take your clusters and try to identify their phenotypes using the tools we have in FlowJo. So I'm going to stick around. So if you have any questions, please feel free to drop them in the Q&A or in the chat, and I'll try and answer them. But yeah, with that, thanks everyone for joining me today. Hope you have a good Thursday. Okay. For those who are sticking around, okay, perfect. Questions? Oh, not a question. So I'm going to go ahead and rename some of these while we're hanging out. This might be a naive T cell, a gamma delta T cell population. This is probably some sort of naive CD8. Um, another gamma delta. Probably on the naive side. HLA-DR positive, CD45, RA negative, CD38. I mean, this is definitely an activated population. I might just call it CD8 active. Okay. So I don't. Okay, here we go. Do I remember? Okay. So there's a good question in the chat about changing the transform to remove like white space or just making the transform good to maximize the separation of the two parameters. So as far as when you should transform your data, yeah, the first thing one of the earliest things I would do when you load your data into FlowJo is transform the data and make sure all the transforms look good, because it will affect your future analysis. Sometimes changing the transform later, we can kind of, you know, save some of the, the previous analysis, or sometimes we can adjust the transform later. But for some things, we need to set the transform before we do it. So we want to set the transform for sure before we do machine learning, because the machine learning will use our transform data. So if the data is not well transformed, it's gonna not separate things well because it's gonna think they're all just one thing. So for instance, again, I transformed all these parameters before I ran it. You'll see some of them are pretty homogeneous, like the PD1, I don't think really worked. But just to show with CD45 here, if we didn't transform it, if we had something more like this, that'd be a pretty bad transform. And if we ran the machine learning, or even we're just looking at our color maps here, CD45, down here in the bottom, right? Actually, you can still see some separation, but oftentimes it will not look very clear about what the CD45 positive populations are. And so, we want to make sure, especially for the machine learning, we want to make sure we set this transform before the machine learning to remove this white space here, and just decrease the maximum essentially to maximize the spread of the positive and negative. And so something like this. Obviously we don't want to, so like for instance, my PD1, there's not much I can do here. It's all negative. I don't think the stain really worked. Some of these other markers as well. So we wouldn't really need to do much with these transforms, but for things where there is a clear positive, negative, or some sort of separation, yeah, we want to separate the transform basically right when we start our analysis, or at least before we do our clustering. And if we do our clustering with a poor transform and then change it later, I'd probably want to go back and redo my clustering with the better transform, because it probably will better separate our data. Okay. So I think hopefully that answers that question. Still 35 people in the chat I can see, or in attendance. So if anyone still has questions, definitely drop them in the chat. I'm just kind of hanging out to answer questions. Though I love spending time with you all. There's a question, are there going to be future webinars about isolation? The answer is I don't remember the schedule off the top of my head. This is going to be recorded on our website, so you can see this one specifically. Let me go to our website real fast. We'll have a future advanced webinar, which will include clustering and some of the stuff I discussed today. You can, of course, most of you probably know this, you can find our webinars under our Learn tab here. So coming up, it looks like we have Intro to BD Research Cloud, Q&A, and then QC normalization. So there's going to be another advanced webinar in the future. I don't know if we're going to have one in May, maybe July. Definitely keep your eye open, but also you can see in the recorded webinars, we just had an advanced webinar in February, for instance. So you can go see the recording of that webinar here. Sometimes they include the data set. I don't know if this one did. No, I'm thinking it didn't. But you can at least check out the recording and see all the tools they're using. They use T-Rex, they use MEM again, and they used Embed. T-Rex is a really cool tool. I would check it out. You can, even the older recordings are going to be basically up to date, high parameter analysis workflow. The fundamentals like flow sum, UMAP, all that sort of stuff hasn't changed, a whole lot in the last couple of years. So these older webinars are still pretty good. Cluster Explorer, we have a whole webinar on Cluster Explorer, which I kind of went through somewhat quickly today. Easy Daffy is a supervised clustering algorithm that can be really neat and really useful. There's actually a few supervised clustering tools that I hope this webinar covers. So yeah, I would check out some of our previously recorded webinars. They go into a lot. I'm gonna close this. Okay. Yeah. And so there's a question about looking at different conditions, like stimulated versus unstimulated. So yeah, our advanced webinars definitely go into a lot more detail about that. So if you look at like that advanced one that was recorded last month in February, the one I just was showing, that includes T-Rex. T-Rex is a really great tool for comparing stimulated versus unstimulated conditions. And so that advanced webinar will go through in detail how to do this. But I guess just to show briefly, I made, when I concatenated the file, two keywords, this treatment and this sample number keyword. The sample number is just to annotate each of the samples, but the treatment is to annotate the different conditions. I have 4 different conditions, healthy controls, COVID, and then one of these, 2 other respiratory diseases. I think one was just the common cold, influenza, and the other one I can't recall. But we can compare these different conditions. If I right click and say create gates on peaks, I can gate these different treatments. I'm going to get 4 different populations here. If I want to see my flow-sum clusters on these different treatments, I can just drag and drop. So here's my healthy control. I'm going to rename this just because that'll make more sense for everyone to healthy. And then this is my COVID, I'm pretty sure. So I'm going to rename this COVID. And we can see maybe some changes in just the major phenotypes between the two. Let's just take a look. So just looking at the numbers, I don't know, we don't see too drastic a shift in any of these percentages, it seems like to me, which I'm actually a little surprised by. But if we then want to look at these same treatments in other ways, I can go to Cluster Explorer. So if I run Cluster Explorer on, yeah, I can run it on this, do Cluster Explorer. I'm going to use the flow-sum populations this time. I'm going to use all my markers except for dead, and just that one. Okay. Oh. It's unhappy with me because I renamed something. Is that why? Flow-sum ungated. I may have to use, I think I have to use this. So that's fine. We can use the flow-sum here. It's that UMAP. Oh, why does it say ungated? Flow-sum ungated. It's getting mad at me because it thinks I renamed it differently. I'm not quite sure why it thinks that. Well, we can look at this one. This hopefully still works. Oops, not flow-sum. Okay, we'll want to remove again the ones that we've already used. Yep. Which one needs? It might be because I'm renaming stuff, and I think it's no longer happy with me because I've done that. Okay, well, let's give up on Cluster Explorer. You can use it to compare different conditions. You can use T-Rex as well, and that's actually what I would suggest using. I just did something here. Oh, don't know what I did. I think I copied it. Well, so yeah, that's some of the ways I'd look at the different conditions. Um, and then of course the classic way you could look at these, whoops, at healthy and COVID. We could always make heat map or make UMAPs as well. So let me just do that. Okay. To change these to UMAPs, I'm just gonna select both of them, double click, and then change this to a pseudocolor plot. UMAP 1. let's do total, which is this one. UMAP 2 for the Y. Okay. Yeah. And then we could always overlay the subpopulations on here. So we can maybe see some differences. Although again, I didn't really see a huge difference in just the expression alone. So I'm guessing we won't see huge differences. Not sure I did that right. I think healthy was on the right. So yeah, we don't really see, I mean, sometimes you'll see really distinct differences between the two conditions. In this case, even though one of these is healthy and one of these is COVID, I'm not really seeing giant changes here between my two conditions. Actually, the things that are disappearing, it looks like, are these little islands that I don't even know what their phenotype is, and we don't really have markers for. But as far as the things that we actually stained for, which are our T cells, I'm not seeing big differences here. I guess maybe there's some depletion down here, it looks like. Tregs will really highlight the differences between these two conditions, though, which I see some other questions. Okay, so yeah, let me go ahead and do T-Rex, why not? Still have 25 people here. So with T-Rex, just to show, because it's kind of neat. Oh, I'm T-Rex loaded. I wasn't planning on using it today, so it's not loaded. T-Rex and because yeah, you can compare these two conditions, and it will really highlight like the little changes here between the two conditions. Other ways we could look at the two conditions, I could look at my CD8 subsets in particular. So I have my total CD8 from healthy. I'm going to put healthy on the right again. Total CD8 from COVID on the left. Even just looking at these, I can see they look basically identical. But we can go ahead and overlay COVID. Overlay healthy. See if we see any differences in any of these like activation subsets. Man, yeah, they're very, very similar, which is kind of interesting. Yeah, sometimes we'll see stronger changes between conditions, but in this case, not much. I wonder if one of our other disease states will have stronger changes. Okay, don't remember what respiratory diseases these are, but they are a respiratory disease of some sort. Pseudocolor plot, unit 1. Right. Just gonna do the same thing I was doing before, which is grab this, drag, drop. And then let's see if there's any change in the major phenotypes here. Well, besides the fact that the color is different, or yeah, did the color is different for this one? Did I put something here? 1, 2, 3, 4. I don't know why I did this one different. Okay, weird. Just that weird. 1, 2. What am I missing? This one has something different. Oh, I don't have the. I see. I didn't grab the total CD8, which I guess I've been including. That's fine. I think. Okay, there we go. Now they're the same. And yeah, as far as differences go, again, maybe some light depletion up here in our, okay, well, this far right one, I guess, probably has the most depletion, it looks like, of uh, CD4 phenotypes as some change in state. So yeah, again, T-Rex can help us maybe really dive deeper in, but if I do some specific clustering on our CD4 cells here, I'd expect we'd see some changes substantial between this condition and the other three. Um, yeah, the other conditions look somewhat similar to each other though. Um, okay. Well, I've gone 35 minutes over now, so thanks everyone who has hung around, but I'm gonna go ahead and probably close the webinar just so marketing doesn't have a super long thing to edit. Um, yeah, any last questions before we close? Okay. If there's no more questions, thanks everyone for joining me today. Hope you have a good rest of your day.